This is an overview of an article entitled A Deep Intronic Variant in LDLR and Familial Hypercholesterolemia, Time to Widen the Scope, published in the December 2018 issue of Circulation, Genomic and Precision Medicine. Familial hypercholesterolemia, or FH, is a common inherited dominant disorder with a prevalence of 1 in 250 and is characterized by high plasma levels of low-density lipoprotein cholesterol, or LDLC. Lifelong exposure to elevated LDLC levels leads to an increased risk for cardiovascular disease. Patients are diagnosed with FH based on clinical criteria, and subsequent genetic analysis is confirmative in a large proportion of FH patients. For molecular confirmation, the well-established FH genes LDLR, ApoB, and PCSK9 are analyzed for the presence of pathogenic mutations. However, no mutations are found in the coding regions of these genes in approximately 5% to 10% of patients with clinically diagnosed FH. Variants in the non-coding region of the LDLR gene have been found in a small number of FH patients, but the clinical relevance of deep intronic variants in FH is unknown. In the paper under discussion, the authors used whole genome sequencing to assess 5 affected and 4 unaffected members of a family with unexplained autosomal dominant FH. In doing so, they identified a novel LDLR intronic variant, which they then assessed in cohorts of FH patients referred for genetic testing. The identified intronic variant was the C2140 plus 103 G to T mutation. Shown here on the left is the original family whose members underwent whole genome sequencing. The mutation was found to perfectly segregate with the FH trait. The authors screened about 3,400 FH patients and identified 8 individuals with the same mutation. On the right is a second family in whom the same mutation was identified by the screening. Here, too, there was perfect segregation of the mutation with disease. In silico analysis with 5 splicing prediction algorithms predicted an increase in splicing signal at a potential cryptic splice donor site consensus, 5 base pairs prior to the identified variant. Activation of this cryptic splice site during transcription would be predicted to result in a 97 base pair intronic DNA insertion and a premature stop in exon 15 of LDLR, as shown here. To assess the in vivo effect of the mutation, cDNA PCR products were generated from RNA of an affected patient from the original FH family. A band of 409 base pairs was identified in addition to the wild-type LDLR cDNA of 312 base pairs, as shown here. Sequencing of the 409 base pair band confirmed the 97 base pair intron insertion between exons 14 and 15, as predicted with the in silico analysis. In conclusion, the identified LDLR mutation is a novel intronic variant that co-segregates with the FH phenotype. It results in a 97 nucleotide insertion, leading to a frameshift and premature stop codon in exon 15 of LDLR. Screening for the variant showed a prevalence of 0.24% in a cohort of FH patients without known FH mutations. This work emphasizes the need to widen the scope from LDLR exon sequencing towards whole LDLR gene sequencing in patients with an as of yet unknown genetic cause of FH.